Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall discuss regulation of metabolism after a meal and more specifically, the secretion of insulin. In previous videos, we discussed how we can regulate metabolism by regulating enzymes, since most of the metabolic reactions are catalyzed by enzymes, and we discussed how we can change the function or catalytic activity of existing enzymes, as well as change the amount of enzymes. So either one of these or both of these will lead to a change in metabolism or regulation of metabolism. So how is metabolism regulated after a meal? The answer is insulin. Insulin plays perhaps the most important role in regulating metabolism after a meal. In this video, we shall look at the first half of this story of regulating metabolism after a meal, and that is how insulin is secreted. In the next video, we'll see the effects of insulin on the human body. So let's see the steps that eventually lead to insulin secretion. During the anticipatory or ingestion phase, you see your food, you smell your food, and you taste your meal. And this leads to activation of your sympathetic nervous system, which then causes an increase in your heart rate as well as your cardiac output. During the digestive absorptive phase, as you start eating your food and the food is being digested as it travels along the GI tract by peristalsis, what happens is there is an increased blood flow to the GI tract via the celiac as well as the mesenteric arteries. This is called gastrointestinal hyperemia. This increased blood flow to the GI tract is gastrointestinal hyperemia. There's roughly about a 25 to 200% increase in the blood flow to the GI tract, which lasts for about 3 to 7 hours. This increased blood flow is associated more with lipid and protein-rich meals. What causes this increased blood flow to the GI system? Extrinsic nerves, local enteric nerves in the GI tract, as well as several hormones that are released by the GI tract, which include vasoactive intestinal peptide, secretin, cholecystokinin, gastrin, neurotensin, and substance P. And these hormones are vasodilators. They make the blood vessels larger at the GI tract, and that leads to increased blood flow. So after a meal, as the carbohydrates are broken down to monosaccharides such as glucose, and the glucose is absorbed into the bloodstream, our blood concentration of glucose slowly rises from about 4 millimolar all the way to about 8 to 10 millimolar. So under normal conditions, the only time when your blood glucose becomes very high, that's as much as 8 to 10 millimolar, is after a meal. So this high blood glucose will, of course, diffuse to different organs, and one of the organs it diffuses to is the pancreas. And there are transport proteins in the pancreas, especially GLUT1 and 3 are important. GLUT2 might play some role, and there may be other transport proteins such as the sodium-dependent glucose transporter that may also play a role. So this is still under intensive research. Previously we thought GLUT2 was the most important, but now we believe GLUT1 and 3 are more important in the pancreatic beta cells compared to GLUT2. Once the glucose is inside the beta cell, it will be phosphorylated by glucokinase. Glucokinase is an enzyme found predominantly in the liver and pancreatic beta cells. Glucose will be converted to glucose 6-phosphate. This enzyme glucokinase does not get saturated easily, unlike other enzymes in tissues such as muscle and red blood cells and so on, because glucokinase has a very high km compared to the other hexokinases. So in this respect, glucokinase is a glucose sensor for the beta cells. As your blood glucose starts rising, more glucose enters the beta cell, is converted to glucose 6-phosphate. So the amount of glucose 6-phosphate that is formed will depend on the amount of glucose in the bloodstream. So now let's see how the glucose that's in the beta pancreatic cells triggers the release of insulin, which will be secreted out into the bloodstream. Insulin secretion by the beta cells of the pancreas occurs by two pathways. One is called the triggering pathway, and the other one is called the amplifying pathway. If you look at the plasma insulin concentration, you see a basal level, basal concentration, and then when you have a meal, insulin secretion is triggered, and you can see this sudden rise of insulin plasma concentration. This happens in the first 5-10 minutes of insulin secretion. We call this the first phase. It's a very rapid phase, and it's also a very robust phase leading to the spike in plasma insulin concentration. So what is this triggering pathway? Let's look at the steps of this pathway that we call triggering pathway. So when glucose enters the beta pancreatic cell, it's converted to glucose 6-phosphate by glucokinase, and then this glucose 6-phosphate undergoes glycolysis, forms pyruvate. Pyruvate then enters the mitochondria, and the PDH reaction converts it to acetyl-CoA. The acetyl-CoA enters the Krebs cycle or the TCA cycle, and that leads to production of NADH and FADH2 that goes to the electron transport chain, oxidative phosphorylation, and out comes ATP. And remember, this is what we called in our previous video the central pathway. So through the central pathway, glucose leads to the production of large amounts of ATP in the beta pancreatic cell. The ATP in the mitochondria is transported out of the mitochondria into the cytoplasm. Now the ATP concentration in the cytoplasm rises, and this ATP now binds these channels called ATP-gated potassium channels. So the ATP binds ATP-gated potassium channels. And this binding of ATP to the potassium channels leads to the closure of these channels. And when these channels close, potassium cannot get out of the cytoplasm. Remember, there's a higher potassium concentration inside the cytoplasm compared to the extracellular fluid because of the sodium-potassium pumps. So as these channels close, potassium cannot get out of the cell. Since potassium is positively charged, this increase in intracellular potassium will lead to increased intracellular positive charge. And this increased intracellular positive charge will cause the depolarization of the plasma membrane. So you can see negative on the inside at the beginning, and now gradually the inside becomes more and more positive or depolarized. This depolarization will cause a change in membrane voltage, and this change in membrane voltage will be sensed by these transport proteins called L-type voltage gated calcium channels, right down here, also called VGCCs. And because of this change in membrane potential, these channels now will open up. Now, normally there's hardly any calcium in the cytoplasm, but when these channels open, the calcium from the extracellular fluid, which is in high concentration, will diffuse across its concentration gradient and enter the cytoplasm. So now we have more and more and more calcium in the cytoplasm of the beta pancreatic cell. Before we discuss the effect of this increased intracellular calcium, let's look at the synthesis as well as the storage of insulin. Like all proteins, insulin is also made in the cytoplasm. The N-terminal signal is synthesized first, and that is followed by the rest of the insulin. This is called pre-proinsulin. Now this pre-proinsulin is then sent to the endoplasmic reticulum, to the ER, and in the ER, the signal, N-terminal signal, is removed, and the pre-proinsulin will become proinsulin. The proinsulin is basically two chains, A chain as well as a B chain, and these two chains are connected together by a C chain, or what we call a C peptide, and there are also disulfide bonds found in the proinsulin. The proinsulin is transported to the Golgi, and in the Golgi, the C chain is removed. The C peptide is removed, and that forms the active insulin. So this active insulin is basically a AB dimer with 3 disulfide bonds. So this is your AB dimer with 3 disulfide bonds. In the next step, the active AB dimer, as well as the C peptide, is packaged and stored in membrane vesicles. Insulin is stored as a complex between 2 zinc ions and 6 insulin molecules to form a zinc hexameric insulin aggregate. The concentration of zinc in these vesicles can reach very high values, as high as millimolar levels. So back to the story of increased calcium. So what does increased calcium do? This increased intracellular calcium is going to bind and activate several proteins such as synaptogamine, and these proteins are involved in the fusion of these membrane vesicles to the plasma membrane. So with increasing calcium concentration, these vesicles that are very close to the plasma membrane, less than 200 nanometers, they start fusing to the plasma membrane. And after fusion, we have exocytosis, and via exocytosis, the active insulin as well as the C peptide is secreted out of the cell. So the active insulin as well as the C peptide eventually would enter the bloodstream. So this is the triggering pathway of insulin secretion. This pathway depends on the increased concentration of glucose in the beta pancreatic cell, which eventually leads to high concentration of ATP that triggers the release of calcium, which causes the fusion of the vesicles and the release of the insulin. The second pathway that causes an increase in insulin secretion is called the amplifying pathway. So while the triggering pathway is of short duration, just a few minutes, the amplifying pathway causes a sustained release, a sustained slow release of insulin. This is the second phase of the amplifying pathway, and this pathway depends on the triggering pathway. That means if the triggering pathway does not happen, the amplifying pathway will not happen. So what is this amplifying pathway? The exact mechanism of this pathway is not clear yet, but basically when the glucose concentration reaches a certain threshold, it triggers this amplifying pathway. And in this pathway, other metabolites have been implicated in causing the exocytosis of the insulin vesicles. Examples of these metabolites include NADPH that comes from the mostly from the hexose monophosphate shunt, glutathione, reduced glutathione, as well as monoacyl glycerol. The amplifying pathway stimulates the fusion of these vesicles that are further away from the plasma membrane. So in order to do that, we have to increase either the concentration of the proteins or activate these proteins that are necessary for movement of these vesicles from deep within the cytoplasm towards the plasma membrane, and eventually then causing the vesicles to fuse with the plasma membrane, resulting in the exocytosis of the insulin. So in this respect, these metabolites somehow increase actin remodeling, affect cell surface receptors, transcription, translation, as well as post-translational modifications of these various proteins that are involved in vesicle transport as well as vesicle fusion. Finally, a few words about insulin in the bloodstream. So the active insulin is a AB dimer. The insulin as well as equal amounts of C-peptide as well as zinc are secreted into the bloodstream. These zinc ions may also act as signals on neighboring beta cells. Clinically, the measurement of the C-peptide in serum samples from diabetic patients allow us to determine if the patient is actually producing any insulin. Because if we do see the C-peptide, then the patient is actually producing insulin into the bloodstream. Insulin is water soluble, so it simply travels in the bloodstream as a dissolved protein. Now, the plasma half-life of insulin is quite short, less than 10 minutes. And part of the reason for this is that the molecular weight of insulin is very low, less than 10,000 Daltons, and anything smaller than 40,000 is very easily filtered into the tubules of the kidneys and eventually excreted if it's not reabsorbed fast enough. Another reason for this low half-life is that insulin can also be taken up by tissues such as the liver, kidneys, and muscles and become degraded. So coming back to our original question, how is our metabolism regulated after a meal? The answer is insulin. So in the first half of this story, we have looked at how this very important hormone is secreted. In the next video, we'll look at the effects of this hormone, insulin, on the body. So until the next video, thank you very much for watching and supporting this channel. Goodbye and have a wonderful day.